Well, good morning, everyone. Welcome to the Informatics Power Talk, now officially under the Department of Biomedical Informatics and Data Science at UAB. Today, we have a featured distinguished speaker from Yale, Professor Hongyu Zhao, to give a talk. And it's hard to suppress my enthusiasm when I introduce him. Because I think Professor Hongyu Zhao is not only hold distinguished endowed position at Yale for Professor of Biostatistics, and also in the statistics department and genetics department, but also because that the vast impact of his research that transcend the traditional biostatistical method development. Many bioinformaticians doing computational biology and bioinformatics may have cited his work, read his paper. His work often goes very deftly between novel statistical methods to very deep, hardcore science, the study of many, many diseases in genetics, molecular biology, precision medicine. And to give a little bit about his background, he has a bachelor degree in probability and statistics from Peking University. I like to be very proud to say that when he graduated, and I just joined in the Peking University. And then he got a PhD from Berkeley in 95. And since 96, he's been doing research at Yale. He have published more than 650 articles and trained over 100 doctoral and postdoctoral students, and many of them are very active in academia. And in fact, before we start, we were talking about one of his first postdoc fellow who was recruited to UAB School of Public Health. And he's very well distinguished and decorated with many awards, including fellows of the AAAS. And he was recognized by the Evelyn Fix Memorial Medal, and cited by UC Berkeley, and also among many other distinctions, including Connecticut Academy of Sciences and Engineering. Without further ado, let's all welcome Professor Hongyu Zhao. I'm sure that he has something really great to share today.
Thank you, Jay. That's very generous introduction. And I still remember I first met you when I was visiting like Indiana, right? I was, I think Keith Dunker was still the chair at that time. Yes. I don't know how many years ago was that.
Yeah, 16 years.
Okay, yeah, yeah. Yeah, very good time visiting. It's really thank you again for kind of invitation. And I'd like to share with you some of the recent in last like recent years, we have been really looking into ways to predict disease risk based on genetics data. And let's see. I think a lot of us like really want to have a crystal ball about our life, our future, especially our health these days. So like not too long after the human genome was sequenced, and also not long after the initial wave GWAS, genome-wide association studies were done, people start asking questions whether we can learn anything beyond just biology, drug targets, and can we just make prediction about our future in terms of whether we have certain diseases or not. And along with that, a lot of direct to consumer companies, and including 23andMe. 23andMe, like the whole business model has been built on providing sort of services to customers about ancestry and also disease risk prediction, although it has shifted the focus to more drug discovery recently. But still, a major part of business is to get people's DNA and to tell people a person's ancestry, and also more importantly, to assess disease risk so that people can be more aware of the potential risk. Therefore, maybe to change behavior and other things to reduce disease risk for different diseases. And so there are just things I can offer that you can get, and you probably see these kits already on a lot of like the drug stores or supermarkets. And so the basic technology that's really driving the genome association studies is been around for 20 plus years. And it got down very, very affordable now. Like, for example, so this is an instrument where it's based on Illumina as Omni 5. So this is a particular chip, can genotype like 5 million genetic variants in the human genome. And also the most commercial companies, they look at the lighter version of the microarray, the genotyping arrays with 700,000 or 800,000 SNPs, or for the all of us, maybe 1 million. But it's very, very like affordable, like each chip, the cost really like $30 or so. So you can really get the person's genotype at the 100,000 size in the genome. If you couple that with some imputation approaches, you can get sort of the variants at millions of size in the human genome. So that gives a very rich, robust way of assessing genetic variations in the human genome. But also, if you combine that with having access to a large cohort of samples, the GWAS is really built on the, having individual genotype and individual phenotype in cohorts. From the first GWAS study with a couple, like 149 people from Yale and Josephine Ho, look at the first GWAS paper looking at age microdegeneration by in 2005. In the last 19 plus years, people really scale up the genome-wide association studies from a couple hundred people to millions of people. Right? So it's just a few. I think some of you may be very familiar with the large like biobanks that are available. The UK Biobank is one of the first in the world. They started recruit participants back in 2006, and they already got more than 500,000 people. They are expanding that. But that has been a great resource for research for a lot of different things for the community. And in the US, there's a couple of programs. One is started a little bit earlier, the US Million Veteran Program, that one of the size for patient recruitment or participant recruitment was in West Haven, not too far from Yale. That's how we got involved from the planning, and also until to this day in the certain project, the US Million Veteran Program. And the goal was to recruit 1 million people. Actually, already passed this goal. So there's it's already got more than 1 million participants, and aiming to get more people. And all of us, also I'm sure all of you know about. And this really initiative started when Obama was in his presidency and called precision medicine. And the goal also still to get more 1 million people. I think this thing that probably will be accomplished in a couple of years, 2026. And on the other side of the ocean, like in the Japan, so there's also available data sets, not population-based cohort, but more like hospital-based cohorts. Biobank Japan with a couple 100,000 people. And also there's a biobank, 500,000 people with like Kaduri, it's a donated by Kaduri, funded by Kaduri from Hong Kong, but with 500,000 people samples. But unfortunately, I think not every single one has been sequenced, and there's still some barrier to get the genotyping down on those. And but in Taiwan, also they have a Taiwan Precision Medicine Biobank. The goal is to recruit 1 million people there. A common thing about UK Biobank, US Million Veteran Program, and also the Taiwan Precision Medicine Biobank is that there's a like national healthcare system in Taiwan, also in UK, and so that you have more like very extensive medical records for those people. So the same for the veteran program. The VA also has like one of the best medical record system in the country, so that you can look into very rich phenotype, not only diagnosis but also imaging, everything else. So it's a great resource. And just in terms of what the people can gather, right, just in terms of how the GWAS is done, it's pretty straightforward. You look at interesting trait, for example, like cancer or prostate cancer, breast cancer, or lung cancer. You get people with cancer without cancer, and you scan their genomes, right? And so there's a lot of columns here that represent the genotypes, maybe a few million columns there. You try to identify which columns on the genotypes are associated with the trait. In this case, you can think of a very simple chi-square test that you can do, and then you couple that with some other creative approaches. You can identify genes or variants associated with a lot of different phenotypes. And GWAS has been very successful, right? For example, like for height, we all know height is very heritable. About 80% of our height is attributable to genetic factors. And also height can be also reliably estimated, right? So if you measure someone's height in the morning, in the evening, there's a tiny bit of difference, but in general, it's pretty robust. And you have a robust phenotype, and also you have a large cohort of people you can get height information. So in this study, a couple year and a half old, they look at height from like 500,000 people, 5.4 million people. They identify like 12,000 sites in the genome associated with the height. So there's a large number of variants, large number of genes involved in height. And in terms of the psychiatric disorders, a lot of genes also have been found. For example, for schizophrenia, and also in this particular study, they identified like almost 300 these different genomic regions associated with schizophrenia. And also for breast cancer, and more than 200, 300 regions have been found associated with the breast cancer. So these are the regions that people have found that pretty confident. There's a lot of other regions that may still be found that may have a larger samples analyzed. So these are all good. The question today is I'm going to really focus on how to turn these large cohorts of people with both phenotype data and the genotype data to estimate the risk for different phenotypes for a trait as simple as height or as complex as schizophrenia. Whether there's a way that we can really, how to make the best use of the genome-wide association data sets to do that. So if you look at the key sort of these, the key issues for any prediction model, you just think about very simple additive models here, right? So I'm going to stick with additive models for today's presentation. Like we look at all the markers for every single marker, and we want to think whether this marker is associated with the trait or not. So the question, which marker want to include? And when the markers included in the model, how to score them, right? So each marker has 3 different genotypes. How to assign the risk scores or whatever scores you think of to the 3 different genotypes. So there's 2 steps, right? There's which markers to choose and how to assign or score each genotype. And you add them up, right? So this is a simple additive model here. So this is the different methods differ in how the markers are chosen and how the individual genotype is scored. And another challenge for deriving risk prediction models, in contrast to a more traditional way of getting the risk or to predict a phenotype, is that we don't have the complete table most of the time. We only have a summary statistics, right? Ideally, for the biobank data, we have individual genotype, have individual phenotype. We can do like different kind of regression models, but a lot of GWAS only provide summary statistics. So what are summary statistics? So suppose we have 1 million markers. So instead of having a table with 1 million columns and a phenotype, we have 1 million rows to summarize the marginal association signals between each of the 1 million markers and the phenotypes. So these are a few rows, and this tells you the genetic variants that you see in the population. This the frequencies. This tells you the effect size, right? So for example, for this one, and the effect size is pretty small, like only to the 3rd decimal point. And the p-value is sort of not impressive. So in this particular case, if you want to assign the come up prediction model for a trait, probably you don't want to use this marker. And some other marker need to be more informative, right? The p-value is 0.003, 8 to the times 10 to minus 6. And also effect size a lot stronger, like a 14, 15 fold stronger than this one. So you may want to include this marker, right? But this is a data set I'll be talking about, we'll be using to come up with to estimate genetic risk, and this actually presents some challenges. Just in terms of the literature for methods to for genetic risk prediction, is a large, extensive, and also still growing literature. Back like more than 20 years ago, by the older days, even before the GWAS, and you can see it really picks up, and it's getting more and more crowded. This is only back to 2021 in a review paper by Zhao and his student. But there's more and more papers coming after this, and also each acronym represents a particular method. Now, all the methods are like colored in two different ways. And this is like blue, and this is more like orange type. And the blue color represent the method that really require or need the input of the individual genotype data. And you can see the most recent methods, they have this color because these methods are based on using summary statistics only. Right? So this is sort of say why. Because when the data get too large, it's probably much easier to analyze or to take in the input of the summary statistic. The other one is like privacy concerns. Especially if you look at like a different cohorts, some people are not willing to share, and also sometimes it's just say like it's just much more difficult to work on individual level data. So there's a difference between variable selection and prediction in statistics, right? So in the most GWAS early days, people want to really identify which genes, what variants are associated with the phenotype. People tend to be very conservative, right? They don't want to make a lot of false positives because you are looking at the millions of variants. And so this is more like the variable selection problem. People want to be very, very sure about what they pick up. But in terms of genetic prediction, and the problem is different, right? The goal is to get as good prediction as we want, and we do, but we don't really care about what variants, what genes are involved in the prediction. We just want to have a prediction model that works. And in this setting, actually, it's quite unique. As in other similar contexts too, but it's very unique in GWAS. Like, even though we have millions of markers for height, we have like 11,000 independent markers associated with height. But even at this very high level of number of markers, this 11,000 markers only accounts for a very small proportion, millions of markers people are looking at. So the signals are still pretty sparse. Not only that, the signals are very, very weak, right? So it's like each marker only increase individuals, even it's associated with the height, tiny bit, right? So the question, you have a very large number of features, only small proportion features are rather informative, and each feature has only weak effect. So this becomes very challenging. And so just to give you, I already mentioned, I'm going to stick with the additive model here. It's one of the simplest model you can think of for prediction, relating the phenotype Y to, I think just now the focus on the continuous phenotype. This X represents the markers you use. There's a P markers, you think about P different single nucleotide polymorphism. And the beta here corresponds to the effect size for the P markers. And so the genetic score is nothing but come up with to really estimate as good as possible the beta, the effect size for different markers, and that's your score. So if you have a good estimate of beta, then we are in a good position to make predictions. And as mentioned, there's many different methods that have been developed to estimate prediction. But think about like they mostly differ how really think about what's good way to specify the prior distribution of beta. Think about biology or what people observe. How do we use that like a properly designed prior distribution so as to derive a good posterior estimate of beta? So different methods just differ in how they specify the priors. And so I'm going to mostly talk about the Bayesian methods because these are the methods that people have found to be more useful than other methods. And different Bayesian methods differ in how they specify the priors. So one of the earlier ones, L.D. Pratt, proposed by Alex Price from Harvard. So they have this very sort of very intuitive prior distribution. So you think about beta, the effect size is a point normal mixture where pi denotes what proportion of the variants have effects on the phenotype. So it's a pi. So when a marker or a variant has effect on the phenotype, they assume the effect size follows random effect follows normal distribution. But for the majority markers, when pi is like 1 minus pi, this is 0 effect. So it's a point normal mixture. And so they design some MC algorithm to estimate. And another method that has recently taken off, also from Boston by Ge Tian and the colleagues, is look at the PRCS. It's another way to specify global local scale mixture of normals. This method computationally is efficient and also empirically has shown very good robust performance. And the software is well designed. People use that more and more. And this is a method that developed by us. I'll mention a little bit more as SDPR. So it's more like think about generalization of the point normal mixture, but generalized in a very generous way. Using the Dirichlet process, we want to say, okay, there's a certain markers, they have no effects. But when the markers have effects, there are some markers with very large effects, markers very small effects, moderate effects. Their effect distribution may not be captured by a simple normal. So we have a mixture of normals. And also between this point normal mixture and our very general one, there's some kind of the SBSR assumed as 3 normals and also other approaches. Okay. And so this is one for our entry into this is we have empirical based method where we just look at point there's a point at 0 and we assume 3 normals, but without really specifying the variance for each normal distribution. We use the data to estimate the sigma, the sort of thing about the effect size for each of the normal component. And so this gives some good results, but the problem for this approach our approach is that we assume the markers are independent, but we know the markers are not independent. So this is something called linkage disequilibrium in population genetics or in human genetics. So this gives you some flavor to look at this. So this represents the genomic region, and every single dot here is a marker. And it corresponds to European samples, like East Asian samples and African samples. And this gives you an idea of the dependence among different markers, right? So in this particular case, you can see this point represents the linkage disequilibrium or dependence between these 2 markers. Dark red means it's highly dependent on each other. You can see among the Europeans, there's this block of markers. They are highly dependent on each other. So the markers are not independent. That's one thing. The second thing I want to note is that between different populations, the dependence pattern actually can be quite different. In general, you'll see much weaker dependence for African samples than for European and also even more so than the East Asian samples. Okay. So extending from the general idea empirical base, but now acknowledging that the markers actually are dependent on each other. And so Wei Jiang in our lab, he has developed something called the GWAP. And the GWAP is a method that really takes into consideration the linkage disequilibrium among the markers, but still use the same the normal mixture model. So in this case, you can see that the prior distribution is still the same, but because the markers are no longer independent, so the posterior distribution, the observed data, the Z, is no longer independent, but rather it depends on R. R is the linkage disequilibrium matrix, right? R more corresponds to how strongly the markers are dependent on each other. And so the Z, whatever you observe, the test statistics will no longer be independent, conditional on beta, but rather they are dependent, depending on how the markers are really related to each other, the effect size. And the GWAP is really based on some empirical base estimate to estimate both the pi as well as the sigma. So going to the other extreme, I mentioned Ge Yu in our lab, he developed SDPR. So SDPR is instead of having just 2 or 3 components, SDPR allows in consideration in principle infinite, but in practice we capped at 1,000 components. And so this is how this is sort of the prior side. And also this is R eta beta really corresponds to whatever Wei has here. But also the variance covariance matrix here is plus 8 times I. And so this sort of the way how we address some of the challenges in the real data set. I think when this was developed beginning, SDPR worked out perfectly in simulations. But when we applied that to some real data and see the performance wasn't as good. And then they figure out for real data are always messier than the simulations. So this is just one example. So this is the two markers. We take the GWAS results from the summary statistics, but you can see the sample size is actually different between these two markers. And also the beta, the effect sizes, the effect size, actually, the effect sizes are in the opposite direction, even though these two markers, they are in strong LD with each other. This is because something funny happened during the meta-analysis. This is not just GWAS from one study, it's GWAS combined result from multiple studies. And so in this particular case, the markers are so strongly dependent on each other. This sort of like really like a weird behavior, and we use this additional like a term here to account for that. Okay. So far, I just give you some very quick overview about like the from the very beginning, like the very point normal mixture model, and to point a mixture of normals and a point of mixture of many normals. And although this can be done or have been implemented, and but so far I haven't really I've been mostly thinking about the markers as random variables, right? It's just a data matrix, but we haven't really taken into account the biology other than we would try to accommodate their physical locations along the genome. But on the other hand, much known is about the functional roles of different parts of human genome. And also much is known about I will talk about the shared genetics across many populations across different traits also has turned out to be a useful thing to have in the business risk prediction. So I've focused on talk about the first part. Just in terms of biology, and so all of us, we have like the 3 billion base pair in our genome, but in different parts of our organs or tissues, our eyes, our lungs, even though every single cell has the same sort of the DNA sequence, but these organs, these tissues actually have different functions because not every single part of our genome is active, right? So there's a very strong genetic control of how each individual tissue, each individual cell types is regulated, right? And so that information also has been gathered by a lot of people in the last 15, 20 years. That information actually can be turned into something we call the human genome annotation. So in this particular case, we can see this particular region, the hemoglobin region, and we can combine a lot of different pieces of information. I'm not going into details, but there's a tools, including this tool divided by us. We can annotate the whole human genome, and in a way that we can make some assessment, make some prediction about a particular part of the genome, whether its genome is relevant or is active in a given tissue or given cell type. So let's look at the tissue we can bring. For the hemoglobin region, we can say this particular region is probably not very relevant, active for functions related to the brain, and not for the GI, but this is a very active, also this is maybe relevant for blood, also for the immune system here. So with such information here, and how that information can be turned into sort of a useful piece to help us to improve risk prediction. So for example, if we're dealing with a psychiatric diseases, for example, schizophrenia, and if we truly believe what we have here, if you think this region is not reactive at all, so we can pretty much ignore, right? So when we do the risk prediction, all these genetic variants in this region, whether they have very strong or moderate or some weak association signals, probably we can maybe benefit by just ignoring or downweighting the evidence from this region so that we can incorporate some of this annotation to help us. But on the other hand, this region may be quite relevant for other blood-related traits or autoimmune diseases or immune diseases. So this is how we can really try to integrate this information there by looking at different columns. Now each column is no longer the same, but rather is annotated with a lot of different pieces of sort of functional information there. We can actually use the data to learn the relevance of different annotations for each trait. And so this is the version like the annual pride along the line of LD pride and developed by Yiming when he was still in our lab. And so basically you can think about how we set up. There's a different way we can set up priors now, taking into account the functional annotation information here. The second prior is very intuitive, saying depending on what annotations you have, the prior for that particular annotation, suppose this is sort of for a given given genetic variant, there's annotations, right? We can know this variant may or may not be relevant in the brain. And based on that information there, we can have different prior to see whether this particular variant is associated with a brain or non-brain related disease. So the point normal this mixture is still valid, but the mixture proportions may be quite different depending on the annotations. So that's one way you can specify the prior. The other way you can specify the prior is like instead of focus on the proportions, we can look at the size. If a variant in a functionally relevant region, we assume the size maybe tend to be larger. There's a different way you can capture it. Or another way you can do it that's implement in the GWAP by way is in this so this very simple link function there, just like this is turned out to be really working out really well. It's saying that so it's just focus on the proportions, but not the proportion pi I is a function of the functional annotations, right? So the pi I, so a given variant, the chunk this variant is involved in the phenotype is a function of the functional annotations. This alpha corresponds to the annotation. This AI corresponds to annotation vector. The vector can be pretty long. This alpha really tells you the importance and relevance of each annotation vector there. So for given disease, the alpha can be estimated from the data, and that information can be injected into this one to improve prediction. And just to show this line of thinking can be useful, so we apply the different methods and compare their performance to the UK Biobank data. As mentioned, the UK Biobank started in 2006, and there's more than 500,000 people there, and they have complete we have very detailed genotype information and also detailed phenotypes, and we can use this as a very good resource to evaluate or compare the performance of different prediction models. And so we look at a bunch of traits on both binary traits as well continuous traits, like asthma, breast cancer. And we use a training data set here. And so the training data set is not the same as UK Biobank. This tells you the size of different training data set. And we look at the testing data from the UK Biobank, for example, for asthma. We have like about 36,000 people with asthma in the database, and about 240,000 people like the controls. And this is sort of the sample size we use to evaluate. So this is for the binary phenotype, and this is for the continuous phenotype. For binary phenotype, just take asthma, for example, in this case, we use the PRS CS as a baseline. PRCS to use a continuous shrinkage is a very common use method on these days. We compare the performance of different methods with respect to PRCS. And so you can see in this particular case, we look at the GWAP and GFNO. So these are two methods that really try to model the linkage disequilibrium information there using the three-component mixture model, and this trying to incorporate the annotation information there. So these two methods seem to stand out compared to the other methods. And compared to PRCS methods, so this SBSR and the very simple pruning and thresholding methods. The pruning thresholding is pruning threshold just more. You pick significant ones, you try to remove dependent markers. It seems to do like a poor job, but in general, the other methods seem to do a better job than the PRCS. So and also you can see across like breast cancer, cardiovascular disease, inflammatory disease, type 2 diabetes, schizophrenia. So across the board, we see that the methods are using these multiple mixtures here. The finite mixtures seem to be doing a better job. And also the more important thing is that this one, the one to the left, the method utilizing the annotation information seem to actually perform the best across different traits. That shows you the benefit of incorporating the functional annotation information here. Also, we see that for the continuous traits, in this case, we can just see that in general, the PRCS is doing a pretty good job, but our proposed method actually seem to do a better job than PRCS. Yeah, and also just in terms of computational time, just see like these are pretty manageable. Like this is GWAP annual. This is a number of like in terms of hours. So this is 0.3 hours for asthma, compared to the other methods. So it's pretty manageable here. and also in terms of memory usage. And the pruning thresholding is sort of least use the least amount, but this is a very simple. It doesn't perform well, and compare on this GWAP annual with the other methods, PRCS, it's pretty comparable. So for the most methods, they are pretty comparable in memory usage. So that's a part talking about the utilization of the functional annotation information really helps, that can push or can improve the prediction accuracy. But the other kind of information we can really borrow, try to integrate, is to use some information on shared genetics. For example, there's a lot of methods as well as applications that demonstrate a lot of shared genetic factors across different traits. This is sort of I'll give you a summary using one of our developed tools to look at genetic sharing from about 55 different traits. And so there's this is correspond to a group of the autoimmune diseases. This corresponds to psychiatric disorders. So in this particular matrix here, and each color corresponds to the magnitude of genetic correlation. And I think the higher genetic correlation means that when they share more, like the effect sizes are more similar across different markers, and the red means the opposite direction. And you can see there's a lot of shared genetics among these autoimmune diseases, among these psychiatric disorders, and also among these metabolic diseases. And also exists some negative correlations there. So if there's two diseases, two phenotypes with a shared genetics, so you can think what very ideal, you can borrow information from, for example, you have a large study for schizophrenia, a smaller study for bipolar disease. If you believe the effect sizes are like correlated between schizophrenia and bipolar, and you can actually borrow some information from schizophrenia GWAS into bipolar, therefore to improve the prediction accuracy. And so along this line, we developed a number of tools and as well as others for improving risk prediction. But more importantly, actually this idea also generalizes, extends to shared genetics between populations, probably even so. Why is it important? So this is a paper that has been highly cited in the GWAS communities by Alicia Martin back in 2019, just to show you some kind of disparity in terms of how the GWAS, the population has been, the GWAS populations like, so this corresponds to the Europeans, and this corresponds to East Asian, etc. But this really represents the proportion of each populations in the world. The Europeans only account for like a small fraction, whereas the East Asians and South Asians account a lot more, but they are not really well represented in the GWAS cohorts. And as a result, if you apply the GWAS prediction models, risk prediction models using GWAS for Europeans, this is the baseline, right? That's how well you can do for the Europeans. But if you apply the model to like the East Asians or South Asians or Africans, you can see the performance actually goes down pretty quickly. So there's a need there. You can for developing like a robust or more accurate risk prediction models for non-European samples. The one challenge I mentioned earlier, I showed the plot. The LD plots can be quite different across different populations. So this also look at the contrast, another region between East Asian, African, and the Europeans and African. So you can see the LD patterns can be quite different. So when the LD patterns are different, it makes it very difficult to really to generalize to make any inference about the causal effects. And when you see this particular case, the causal effects actually are the same between the two populations. The observed signals can be quite different. But on the other hand, when the causal effects are different between the populations, the observation can be similar. This is all due to that very different LD patterns between populations. So if you look at the results from one population, another population, if you do consider the LD, the linkage distribution pattern differences between populations, that can lead to some misleading results. And so along this line, by explicitly modeling the population specific linkage disequilibrium matrices, also taking into account the population specific GWAS results, you develop STPRX. So this is more like the cross population model, where we think about this way. We try to borrow information of the shared genetics across. This is how this is done. So all the markers are partitioned into 4 different groups. So there's a group of markers that has no the genetic effects on the given trait for both populations. There's a one group markers that only has effect in one population, the first one. There's a one group markers only has effect in the second population. Also, there's a group markers they have effects in both populations. For those markers, there's a key parameter here, rho, that corresponds to the genetic correlation, the similarity of the effects between populations. So this rho is a key to allow you to transfer some of the knowledge of the effect size estimate from one population to the second one. In this particular case, you probably have more samples for the Europeans, therefore you have more accurate estimate. For the smaller samples, for example, the Hispanics or African Americans or Africans, you don't have as good, but you can use some information from the Europeans because of the rho, the shared genetic effects. That's the genetic correlation there. Okay. And the rho is estimated from this method called popcorn. And this is how the little bit detail how the algorithm was implemented. And also we compare our method to like a PRCS-X. So X corresponds to cross-population version of using continuous shrinkage prior. This method, even though tries to large information across populations, but doesn't really capitalize on the genetic correlation of the effect size in this method. LD-Pri2 is a single population-based method, and so these are like some based on cross populations, some based on single populations, and then we also look at a number of traits. This is sort of correspond to the training data set, right? First, these traits, these are European samples available across, and some traits we have the most traits here. We have the East Asian samples, but smaller sample sizes. For African samples, we only have the GWAS results for a subset of traits. So the idea is we take the GWAS summary statistics from the training data sets, and we develop model, and we see how these models perform on the UK Biobank, because UK Biobank not only has European samples, but also has like East Asian samples, South Asian samples, as well as African samples. And so this is a busy slide that will show the performance of different methods across different traits. Like for example, for height, this STPRS. So this is a performance based on our method. And the second one is PRCS-X, and follow the other methods. If you look across different traits for height, BMI, HDL, LDL, our methods actually seem to be doing a better job than the other methods. And also for binary traits for type 2 diabetes on the coronary artery disease. We think the key that our method doing better job because our method really makes very explicit use of the genetic correlation between populations. And this is for East Asian, and this is for Africans, the same. And also we can combine this with individual population data and to get even better estimate. And but in addition to this, like we can think about this different population, like we talk about the East Asians and also the Europeans, Africans. But in the US, as a lot like a lot of the population, especially for African Americans, they are not just pure Europeans, they're not just pure Africans, because a lot of these individuals, they are actually as a mixture of European genome as well as African genome, because there's like there's marriages between European samples and also the African individuals. So as a result, in the African Americans, there are maybe up to 20% or even higher the percentage of the genome is from the European ancestry. For example, President Obama, 50% his genome is European, 50% is African, right? So you can't just look at every single genome. You can have a way of inferring the local like genetic ancestry. And for those individuals there, so therefore the prediction actually not only we need to model, there may be benefit of modeling both the genetic markers effects as well as the local ancestry effects. And so this just think about this is one way to think about how the individual genome, how you read along. So these are the DNA sequences, and but also each chromosome also partitioned into different segments with different ancestries. And for ADMIS individuals, you can model the phenotype as an additive effect, it covariates, and also the genotypes. Now the genotypes not only just 0, 1, 2, but also depends on the local genetic ancestry. So we can actually have a way to estimate ancestry specific effects, and that can be incorporated and to have given the time, just saying to have a more accurate estimate. So this is our method, SDPR admixture, still built on Geyu's SDPR framework. And so this is a Gaudi method. This is a paper recently appeared in, I think, Nature Communications. That's looking at also is admixture based the prediction. And I can see, you can see that our methods that really make some explicit modeling of the local ancestry and through this sort of the Dirichlet mixture approach seem to be doing a better job than this one. Okay. So in the last few minutes, I want just to give you sort of a couple of examples. Going into it in depth, we talk about the phenotype very broadly, like the breast cancer, asthma. But I haven't gone into detail look at individual, but to see how this can be applied, how the PRS looks like in the population, how can really inform us to identify people at higher risk for diseases. So this is a pipeline that we use to look at the breast cancer in the UK Biobank. So in this particular case, there's a total about 150,000 females. Among them, about like 9,000 females actually develop breast cancer. So this is on the way on the sort of testing data set to assess for us to really look at evaluate the performance of different risk prediction models. So in this case, we compared performance of different risk prediction models. We use annual pride. So this is earlier work than the GenoWeb, the GWeb annual. So the annual price seems to be doing a better job. Annual price is a version also using annotation information, but built on this point normal mixture model, not point like multiple normal mixture model. But it's overall is also doing this a better job. And also we can, this is how the PRS looks like in the UK Biobank. So there's a two group of people. The controls correspond to those females who didn't develop breast cancer at the time that the data was accessed. And this case is about 9,000 people who had already developed breast cancer when we were looking into the UK Biobank database. And you can see there's a global shift to the right for those people developing breast cancer, but there's lots of overlap here. So there's a lot of overlap between these two cohorts. Another way to look at this, so instead of thinking about the large overlap, but really partition on these 150,000 people into 100 bins. Now each bin has about 1,500 people. So we run all these bins from the individual with the lowest PRS to people with the highest PRS. As you can see here, for people with the highest PRS, it's 1,500 people here. About like 17% had already developed breast cancer when we were looking at the data here. And it is in comparison to people, the lowest bin with the 1,500 people, like there's only 2 or 3%. So between these two, there's a fold difference. So for the chunk of the people in the middle here, you couldn't see much difference. I mean, so there's pretty much like a population level. But for people in especially at the high risk end, we do see some increase of the risk for developing breast cancer. It's already ongoing trial, like wisdom trial, to incorporate genetic risk predictions to see whether what information there can allow for more effective monitoring of the breast cancer in the general population that can lead to some reduced sort of the mortality by early treatment. Also, I know there's ongoing effort for prostate cancer as well as for lung cancer to utilize the PRS. But that's only the one side of the story, right? That we said was for people working in breast cancer, like before the genetics time, people already know there's a lot of risk factors for breast cancer, and there's having to do with the hormones and the age, and as well as the childbirth history. So all these things also relevant for also family history too. And so the information can be also we went into the UK Biobank and we look at a lot of different factors there. And we identify a few factors that have also these clinical variables that also associate with the breast cancer. Now the question is, in order to really get a good prediction, not only do we want to incorporate genetics, but also other available information there to get the most accurate prediction here. And then we compare 3 different models here. And so this one is to use the clinical information. We can get AUC 0.6, so there's information there. For the PRS method, the prediction based on the genetic risk, genetic information there actually is higher than when you combine all the clinical information there. Actually, it's remarkable. But if you combine the two, actually the AUC can be approaching 0.7. That tells you there's some maybe some overlapping information here, but definitely there's a complementary just in terms of getting the most accurate prediction of the cancer risk for breast cancer. And this tells you if you group these different people in the UK Biobank, look at their sort of age of onset, you can clearly see the difference. Also better separation the different these different groups if you combine the genetic information and also clinical information there. And also we look at how these PRS, these genetic risk factors can be modulated or can be moderated by the behavior, the lifestyle factors. So in this case, we look at like type 2 diabetes and coronary artery disease and a few other traits. I'll just show the results for type 2 diabetes here. If you look into the UK Biobank, we can look at a person's body mass index and the exercise and also the diet as well as the smoking, right? So based on these 4 factors, we can classify each individual into like someone having ideal lifestyle, like someone who has really good diet, doesn't smoke, does regular exercise, or someone like poor lifestyle, who smokes, who eats a lot of red meat, and who has a very high BMI. And if you look at the 3 different groups, the majority is more in the intermediate range. So this corresponds to 3 different groups. For each group, there's a different bins or different subgroups where these individuals are further partitioned into the genetic risk levels. Going from left to right, you see the people from left to right, they tend to have a higher genetic risk. So for the same lifestyle, you can just see that the higher genetic risk also makes things even worse. But for people with the same genetic risk, better lifestyle actually reduces the people's risk for disease. That's saying there's intervention that can be implemented to reduce even for people who have a higher risk of like genetic based on genetic load. If you look at the people with a high genetic risk with like ideal lifestyle, so this is the risk level here. And this actually, if you go all the way here, and actually the risk actually is lower than people with a very low genetic risk but poor lifestyle. So just give you some idea about how the lifestyle and also genetic factor they can interact. Also realizing that the lifestyle also can be genetically influenced too, so it's not totally complete, a separate story. Okay. Just to summarize, I've gone through some of sort of the basic ideas across the different methods, how people have tried, including us, to really utilize this Bayes idea to have a proper way to specify the prior information for effect sites across all the markers, but more importantly, how to incorporate genetic annotation information there, how much we know about the genome, the functional regions of the genome to improve the risk prediction, as well as to leverage on the shared genetics effects, more like for the same variant, it may have similar effects across different traits, also more important across populations. That's especially important when we try to generalize that when you combine data from multiple populations, especially for populations that are understudied. And we say that marker dependence can benefit, and also the functional annotation also can benefit. And also I'm talking about the random effects model being normal, but also people have proposed other models to quantify the effect size, to characterize the effect size distribution there. And also the old sort of discussion has been focused on summary stat. But when you have individual level data, definitely you can do better, but how much better? And people probably there's something worth studying especially when you have a individual level data for a minority population there may be potential gain. And also, I want to talk about the risk prediction using for the genetic information, also the clinical information. But when we have medical records, not only we have one time like a snapshot of the clinical information, but also we have the longitudinal measuring information. That information also can be quite useful, especially on the individual-based risk prediction. And also there's a lot of room there when we have the whole genome sequence data, like for all of us, like these people will be whole genome sequence for UK Biobank. Every single one have been whole genome sequence. You have identified a lot of like rare, extremely rare or unique variants. How to incorporate those rare variants is not a challenge. In the last slide, I want to acknowledge the people who really did the work that I'm presenting here, and by Wei Jiang as a research scientist in my lab. Ge Yu is a postdoc, Yu Xuan and Yi Ming and Xun Shi, they were all students in the lab. And also this would not be possible without having access to this very generous resource of the UK Biobank and a lot of other cohorts from NIH through the DBIDS. Also, I'll stop here and thank you for your attention.
All right, can you hear me, Professor Zhao?
Yeah, I can hear you. Yeah, thank you. Yeah.
Yeah, I changed to the on-site location. So I think it's very impressive work. Yeah, we have some people in the audience, and I'm going to ask questions. Chen, yeah, go ahead.
Hi, Dr. Zhao. So we met last year's ASHG meeting, if you recall.
Oh, yeah,
Yeah,
thank you.
yeah, great talk. Thank you so much, and thanks. I was thinking you will be on campus, but yeah, it's still very impressive. So I have a naive question regarding I don't know if the allele frequency play an important role in terms of the accuracy, and in the last slides you mentioned the rare variants and even de novo variants, right? So how to incorporate the rare variants or de novo variants, or the allele frequency that have lower allele frequency variants into the model and have better prediction?
This is a great question. Yeah. So in the setup, all the genotypes are standardized. So that the implicit assumption that for the variance, right, the variance of the effect size is inversely proportional to the minor frequency times 1 minus p. So it's very implicit in that standardization there. So that's incorporated that way. But LDAK model said, no, that's not quite accurate. Maybe that should be a function of p times 1 minus p. Also, there's LD pi. So people are getting a little bit elaborate, but in general, the current setup it's already taking into account of the allele frequency through the genotype standardization there. But for very rare variants, but lots of function variants, and I don't really know what's the best way. It's an empirical question how to do that. But people are starting to look into it. Yeah.
Yeah. So Hongyu, since you and I know each other for a long time, and you know that you're such a rare intellectual, make great contribution to the field. So I'd like to ask hard question, both on behalf of faculty and maybe biostatistics faculty following your footsteps, and also on behalf of students, right? So get ready.
Just interject for really quickly, Jake, sorry. Just we're at 11 o'clock now, so we're officially at time. There's still many people who are still on the call, so thank you for sticking around to the end. And if you have time, stick around for questions. If not, then please don't feel like you have to stay. But I'm glad to see all the great interest. So go ahead, Jake.
Yeah, if you stay, and you'll be rewarded, because I'm going to ask some controversial questions, right? So I think you've been in statistical genetics for as long as I can remember, and I would regard the 2000 to 2020 as a golden age for GWAS and statistical genetics, and you pioneered just a polygenic risk score over biobank data. That's very impressive. Now we're moving into a new century, a decade, and where deep learning models are becoming more impressive, right? So you can imagine many of these data, you don't have to select features, and then you can use deep learning, ingest all the data, and then put together maybe clinical information and other information, perhaps get better AUC performance And it seems to be a trend that's worrying to many followers about statistics, that perhaps moving into a black box deep learning approach and then interpret it later seems to be the solution. So in your opinion, what is the remaining frontiers of statistical genetic work? That's for one. And also, what is your suggestion to people developing their career in this field? Yes.
No, this is a good, I think, great question. The first one that that's come up pretty much for every single presentation I gave, like the deep learning, right, for PRS, for risk prediction. I think this is the case where I actually does really see the benefit of statistical modeling than deep learning. I not only one, but actually multiple people in my lab, they have come to me saying, I want to apply deep learning to GWAS risk prediction. And yeah, sure, they tried like different architecture, different things. There's no method like repeating statistical approaches, right? Because I think it had to do with the unique nature of these the GWAS signals. They are, let's say, very sparse, very weak, and deep learning methods are very good to pick up signals when you have like moderate or strong signals. There's a lot of interactions, nonlinearity, but these things do not apply in this particular context. So there's still room or place for PRS based on this Bayesian approach. But if you think about like now, if you consider like clinical variables, other things, that's a different story, right? We haven't explored, but you might think, but still, I haven't seen convincing evidence that deep learning based from medical record coupled with actually giving better performance than what. So this still time will tell, but purely based on genetic, I think that this Bayesian approach will still be the sort of will prevail. So as to what will need to be done, or there's still room for GWAS, I remember like even like more than 10 years ago, 15 years ago, one of my postdocs, when she was interviewed at Rutgers, and one faculty like in the audience, I say, I just read New York Times article that even by time they called it the death of GWAS at that time, right? Saying okay, given so much investment, not much has been found. I mean, there's no biology, there's no drug targets. I think despite that, people are still making progress. There's a lot of interesting things being found. But also, we also have been, as you know, doing a lot of like also other functional studies. Okay, single cells coupled with the genotype, eQTL, other things. I think this will combine, will give us a lot more interest. Even another thing that's not in sports space, horizontal sequence data. And people a lot have been analyzed, but in my view, those data have been really analyzed to the full extent. So there's all different angles, you can take on to really understand what's going on, and also if you combine that with the imaging data, other data sets, variables. So there's a lot that can be learned for our genetics and also epigenetics too.
Dr. Zhao, Jim Semino, Chair of Biomedical Informatics. Thank you so much. If you can stick around and answer other questions if other people have them. I have to go, but I did want to take a chance to thank you very much for contributing to our seminar series.
All right, thank you. Yeah, nice meeting you, Jim. Yeah, I appreciate it. Yeah, thank you for yeah.
Okay.
Thank you.
All right, take care.
Thank you to you.
Yeah, we have one question from the inbox.
Randers Sminsky. Just got a quick question. So PRS, that's polygenic risk scores. And my understanding is that these are a combination of SNPs into a single score to predict the disease risk. So would I be correct in this data?
Yes, yeah.
Sure. To be clear. And my question is, where do you see direction finding out polygenic risk scores? Because I know currently I'm doing research in Gallup and finding out like the PRS and like finding out different SNPs into different risk scores. So someone will just say like has because with complex gene traits with there are many SNPs and not just one like will. So what do you see going in the future? Like let's just say you find several SNPs and someone has a risk of gout. So how would you like address the interpret it so a patient can understand this?
So I couldn't hear clearly.
Jake, can you repeat the question or at least summarize?
Jake, can you just summarize? Yeah, I just echoing somehow. Jake, I couldn't hear clearly. Yeah.
Through the microphone. Yeah. I think it's a little far away.
Yeah, sure. Sorry. So my question is with the polygenic risk score. So if someone has like, let's just say a risk of gout, so let's just say they have, like, how do you like use this and like interpret to like the the patient? like if they have a risk, like let's just say they have like certain like intermediate risk scores, because it's not just one SNPs, but several SNPs and they're different expressions. So how do you like present to like patient or doctor that someone has a risk of gout, how to address that?
No, I think you've actually very important question, like in terms of clinical usage, right? So at the population level, these scores can identify people with a higher risk for certain phenotype. You can do, like I mentioned, the monitoring and screening. If you come an individual patient or suppose already has gout or other phenotypes, yeah, and then the score, I think we have been working with as a cardiologist, Pradeep, Natarajan, and like Mass Jan. So we are looking at different ways to partition the score. So if someone already have a high score for, someone already has a disease, we may look at the partition the score into different components, and we saw that when we look at different components, there are certain components may have to do with how people respond to different treatments, right? So there are certain things that may give you some, but it's because overall the score, depending on the trait, the correlation there may still be quite low. So the clinical usage, if you already have some disease, may not be as high, but sometimes can still give you some hint. It's more like when you have people with sort of a childhood disease or newborn congenital disease, people done a lot of whole exome sequencing these days, whole genome sequencing. Sometimes you find the culprit, sometimes you end up with nothing. So it's the same setup here. It really depends on if someone may have very low score, still has a disease, then what? Yeah, I'm not sure if I'm not answering your question. No, yeah.
Just taking all this and just interpreting it, because it's one thing to have the data and just interpreting and presenting and.
Oh yeah, I think the term presenting this one that may. That I think still people a lot of work like had to be done to really to see how it can be translated into useful thing to communicate between from physician to the patients. Yeah, it's a long way to go. That may or may not. Also, you need to also consider other covariates too, the clinical variables as well, not just PRS. It's only one piece of information you use. So.
Even when PRS was also raised and several factors. But thank you.
Right. Yeah. Sure. Yeah.
Have another question?
Also,
Yeah.
feel free to raise your hand or post your questions in the chat, or you can unmute yourself too.
I have a quick question.
Yeah.
So Dr. Zhao, if I'm correct, the majority of the GWAS studies were focused on SNPs, right? Single nucleotide variants. So I'm working on stratifications, and there's a small indel of the stratifications. What's the reason that these type of variants weren't? So these variants can be applied in this model as well, right? Why they are not studied as extensive as SNPs?
No, I think a couple of things. One is that the structural variants are these indel, they haven't been traditionally analyzed in GWAS, right? You don't have much sort of association like a summary stat for those. The second thing is people have also found some of the structural variants and the indels can be tagged well by the single polymorphism. So even though you are really not looking at the structural variants or indels, but these SNPs can be a good proxies, maybe. But if you have more direct evidence, certainly, if you have very large structural variant, you know it's going to have some profound effect, and then that's going to be taken into account. Right, but so far it's only what the data we have. We only look at SNPs. Yeah.
Thank you.
So another question that I have about is your breast cancer polygenic risk model. Did you compare the high risk and low risk actual SNP set to determine what are causing it? Is it DNA repair P53 or whatever the thing? And then what are the actionable things that you can do to women, since today is Women's Day? And then what are the things that you can do to women so that they don't go actually into the breast cancer incidence?
No, there are two aspects. One is that for the prediction there for breast cancer, we probably use tens of thousands of SNPs, right? So I try to make distinction between variable selection for more the GWAS, focus on the discoveries versus prediction. The prediction, the goal is to get prediction models. A lot of these variants used may have nothing to do with breast cancer, but they are included because we don't want to throw out some signals. So we have to get a balance between those. So in the PR setting, like the markers used may or may not have anything to do with the GWAS, the breast cancer onset. As in terms of the treatment, that's a very different question. Like I also been working on more on the other side, the somatic mutation side of breast cancer. If you look at the overlap or between the signal for the germline variants and also somatic mutations, actually a student might like wasted maybe a year or more and couldn't find any overlap. So that's the question that still needs a lot of research to really see how these GMAs, the germline variants, are related to somatic variants and how. But in terms of treatment, the somatic variants of the breast cancer subtypes are more dictated by those somatic variants. So the PRS may become probably less relevant or irrelevant in terms of how a patient is treated, at least as far as I know. Maybe there's a way, maybe in the future it's found out this germline variant may be factored into it, but right now I say the treatment decision is mostly based on the molecular profile, on somatic mutations, gene expression patterns, and other things.
That's Amy Wong. I have a question. Would you elaborate on the slide? You don't have to show it, but on the slide where you show the different interrelationship or dependency of the different of the different genes.
Which one?
Based on the different ethnic groups. Could you elaborate on that? I'm wondering, how much of that can be attributed to, well, obviously not because it differs with different ethnic groups, but how much of that can be attributed to co-occurrence and proximity, being on the same segment or very in close adjacency versus other mechanisms for those dependencies, like maybe depending on certain biochemical pathways or things like that?
That's a good question. Yeah. So one thing that people have found out, I mean, there's a couple of recent papers. So there are different ways to look at for given variant, right? If a variant in different populations, whether this variant has very different effects or similar effects. And what people have found more recently that it seems like regardless of genetic context, the ancestry, the biology is similar across different population groups. So the effect size may be quite similar. The thing that's making it so difficult to really translate the GWAS from one population to the other populations is that it's a dependence structure there, because what the signal we see, we are not really probably seeing the real biological signal, rather the proxies or the surrogate markers that correspond to the genetic, the variants with the true genetic effects. So the LD differences across population may account for a substantial part of the failure or the lack of transferability of the PRs from Europeans to non-European populations. But if we really has our hands on the truly functional ones, what people have found is that the effect sizes do not differ too much across populations. So biologically, we are the same Homo sapiens.
Okay, so I see. So part of it could just be that those populations diverged, and so those different variants have just been inherited together, co-inherited by those certain populations. But still, that's something that you have to account for in your model when you say, oh, is this correlated? Does this affect things? So, okay, thanks for adding to that.
I think it's more the statistical dependence, not the biological the differences. Yeah.
So I'm sure that there will be more interest, but then we already tie you up for additional 15 minutes. We want to thank you again, but hopefully maybe this is a justification for the future. We'll have in-person visit and we can.
I would love to. Yeah, I'll be here. All right, thank you so much for having me. Yeah, thank you for all the questions.
Thank you very much.
All right, thank you. Yeah, okay, bye-bye. Yeah.
Okay,
bye.
Thanks, everyone. Have a great weekend. See you at the next PowerTalk.
Happy International Women's Day, as Jake said. Bye-bye.
Bye-bye.
Bye-bye, yeah. Bye.